Today, we are diving into something truly fascinating: how scientists are basically hacking tiny microbes, turning them into super efficient factories for making greener fuel. We're talking about hijacking their internal machinery to produce ethanol better and faster than ever before. This, my friends, is the incredible world of metabolic engineering. So here's how we're going to break it all down. First, we'll tackle the big fuel problem we're facing. Then, we'll look at this stubborn raw material that holds all the good stuff. From there, we dive into how scientists are redesigning a living factory to create what I like to call an ethanol supercell. And we'll finish up by looking at the amazing results.
Okay, so first up, why are we even talking about ethanol? I mean, it's not a new thing, but its role in our energy future, yeah, that's becoming more critical than ever.
And that really gets to the heart of it. We're not just talking about an additive for your gas tank. Ethanol has the potential to be a full-on replacement for gasoline. It's a biofuel, which means it comes from renewable stuff, and it's a way cleaner alternative to the fossil fuels we're so hooked on. But here's the catch, the big bottleneck. The idea is fantastic, but making it work financially has always been the tricky part. You see, to really take on big oil, you have to make ethanol on a massive scale, from things that aren't food, and you've got to do it cheap. That's the real puzzle we have to solve. So if we're not using corn or sugar cane, what's left? Well, the answer is literally all around us, in things like wood chips, corn stalks, all that agricultural waste. But this stuff, called lignocellulose, is a major scientific headache. And that's the million-dollar question, right? All that energy, all that sugar we need to make fuel is trapped inside the tough, woody parts of plants. Prying it loose efficiently is the first giant hurdle to making a biofuel that's actually sustainable. And this diagram really shows you it's not a simple process. You can't just toss some wood chips in a tank and get fuel. No way. First, you have to bust open that tough lignocellulose structure to free up the simple sugars inside. Only after you've done that can you get to the fermentation part. And when you look at what it's made of, you can see why it's so tough. It's a real jumble of stuff. You've got cellulose, which gives you glucose. That's the good stuff we want. But then there's hemicellulose, which gives you all these other kinds of sugars, and lignin, which is basically like a tough glue that microbes can't digest. It's a messy, complicated starting material. So with this really complex problem, scientists knew they couldn't just hope for the best with what nature gave them. They needed a whole new playbook. They needed a way to literally redesign biology to do their bidding. And this is where metabolic engineering makes its grand entrance. This right here is the absolute key to everything we're talking about. We're not just finding a microbe that happens to make a little ethanol. We are going inside the cell, right into its DNA, and reprogramming its internal pathways to turn it into a specialized machine that makes one thing and one thing only: our fuel. You know, the factory analogy is just perfect. A normal cell takes in sugar and uses all these different assembly lines, its metabolic pathways, to make everything it needs to live. Metabolic engineering is like a new manager showing up, shutting down all the unnecessary assembly lines and diverting all the raw materials and energy to one single, super-powered production line for ethanol. Okay, so let's pop the hood and see how this rewiring actually works. For this job, scientists picked a very common bacteria, E. coli, as their microscopic factory floor. Now, take a look at this map. I know, it looks super complicated, but just focus on that one molecule in the middle: pyruvate. Think of it as a busy traffic circle inside the cell's factory. From this single point, the cell can naturally go off in all sorts of directions to make different things. And for our purposes, that's incredibly inefficient. So here's the problem in a nutshell. A regular, off-the-shelf E. coli cell is basically a leaky factory. It takes the sugar you feed it and produces this whole cocktail of chemicals. Every bit of acetate or lactate it makes is a bit of sugar that didn't become ethanol. It's all just waste, and it's what makes the old process a non-starter financially. This is where things get really clever. Instead of just working with the cell's default settings, the engineers decided to give it a brand new instruction manual, a direct, non-stop, high-speed expressway straight to ethanol. And they did this by installing 2 new genes, borrowed from another organism, right into the E. coli's DNA. The first gene, PDC, creates an enzyme that's like building a brand new, high-speed off-ramp from that main pyruvate traffic circle. The second gene, ADH, adds the final piece of the puzzle, paving the rest of the road to pure ethanol. It's a brand new biological highway. And this just lays it all out, plain and simple. Before, that pyruvate could wander off down any old road, making a mix of stuff. But after the engineering, that new ethanol superhighway is built, and pretty much all the traffic is forced down that one super efficient route. No more detours.
So the idea sounds amazing on paper, but did it actually work? Let's get to the payoff. Let's look at the data and see just how powerful this genetic redesign really is.
I mean, just look at these numbers. They are absolutely wild. On the left, you have the original E. coli, which under these conditions made basically zero ethanol. It was too busy making all this lactate and acetate. Now, look at the right column, the engineered one. Ethanol production shoots up from nothing to 482. And those wasteful byproducts, they've practically disappeared. This one number says it all. Over 90%. More than 90% of the sugar that the microbe eats is now funneled directly into making the one thing we want. That leaky factory? It's been completely sealed up. The new assembly line is running at almost perfect efficiency. This is the kind of breakthrough that can make biofuel from waste a real thing. And that leaves us with a pretty mind-blowing thought, doesn't it? We've seen how scientists can literally reprogram a living thing to do a specific job with unbelievable precision. So if we can design a microbe to be a hyper-efficient fuel factory, what other massive challenges could we solve by redesigning life itself?